this computer. Okay, cool. So today is lecture 5, and it's the second half of modeling biological sequences with hidden Markov models. So just to recap where we were. So what we did last time is that we covered HMM basics, evaluation, parsing, and posterior decoding. We looked at observations, models, Bayes rule, and Bayesian sequencing and Bayesian inference. Basically, the key idea is that we're now going to be able to model rather than just simply align. And the way that we're going to be modeling is by having 3 tasks, which were the rows of our matrix. The first task is the ability to emit DNA sequences of a certain type. So basically creating sort of emissions from different classes of elements. The second one was to recognize DNA sequence of a certain type. So now if you see the sequence, can you infer what is the hidden state that most likely gave rise to it? And today we're also going to be learning about how to infer the probability distributions of those. And what we did was to separate the hidden from the observed. And then the hidden was the model of the inferences we make about the world. And then the observed was, you know, what we actually observe. And we looked at how we can use Bayes' rule to effectively reverse the arrow of directionality of those conditional probabilities. Instead of saying, what is the probability of the data given that hypothesis, which is what our generative model gives us, we basically wanted to know what is the most likely hypothesis given the data. And we did that using Bayes' rule, where we calculated the posterior probability using the forward probability, the likelihood, and scaling that by the prior, and then dividing everything by the evidence or the marginal, so P of D. And then we did a very simple proof of Bayes' rule, namely, given that P of A given B, A intersection B can be given as A given B times P of B or B given A times P of A. We can just infer P of A given B as a function of P of B given A by scaling by P of A and then dividing by the marginal. And then this is the actual numbers of how it works out. What HMMs did is couple this Bayesian inference with a Markov chain at the top that govern the transitions. And then we talked about the X vector of observations and the pi vector of the path or the parse, the hidden interpretation of the observed emissions. We talked about this emission vector, which basically is giving us for every state the probability of emitting each of the characters. That's the probability of a given character given the state. And about the transition probabilities, which is given the previous state at position I minus 1 is K, what is the probability with which the next state will be L, okay? And then we saw a very simple hidden Markov model detecting background sequences versus GC-rich promoters based on differences in their nucleotide frequencies and differences in their transition probabilities between them. We saw how we can calculate the total probability of that model given a path and a set of observations, and how we could basically parse this sequence, ATT, AGG, TCTA, in 2 ways, either all background or all promoter. And we saw that all background gives us a probability of 5 times 10 to the minus 9. All promoter gives us a probability of 2 times 10 to the minus 9, and therefore the likelihood ratio is indicating that this is much more likely to be all background. We also saw that the cost of parsing as background here and then promoter here and the background here in order to capture the GC richness was in fact not worth it because of the very low probability of making 2 transitions within 4 steps of each other, giving us a much lower probability. We then saw that calculating each of those would take forever because there's an exponential number of possible paths, and we defined the Viterbi algorithm that allows us to now infer the maximum likelihood path over, taking the maximum over all possible paths, and then selecting the best path. So this is a single path column again, where we're choosing the most likely path. And how did we do that? We did that using dynamic programming by defining this Viterbi variable that allowed us to express the maximum probability over all possible paths ending at that state as a function recursively of smaller subproblems, and the smaller subproblems were the previous time point. So basically, given that I know the maximum likelihood score of ending at each of the states in the previous time point, I can now multiply those, scale those by the transition probability, i.e. the compatibility between state J and state K, i.e. how often is state J transitioning to state K. So I'm scaling the maximum previous state probability that I could possibly obtain over any path ending at that state, scaling that by the transition from the previous state to the current state, and by the emission probability. And then using all that, I'm now defining the next maximum variable. This is the maximum score that any path ending at state K at position I can possibly have. And that basically defines Viterbi recursively as a function of itself, enabling us to now compute it from the left to the right. Okay, so let's see who's 100% with me so far based on the sort of review of the previous lecture. So refresher of HMMs. This is awesome. So 19, 10, 0, 0, 0. This is really great. Okay, so that was Viterbi. And then we also defined the forward algorithm that gave us the total probability summed over all paths. Okay, so now we enter this mysterious second column, and this was again scoring of a given sequence, but now scoring not just over one path, but over all paths. So that's basically not just the probability of X given a given path, but the total sum probability of all of that, summing over all paths, basically marginalizing over all paths, and then ending up with the total P of X regardless of the path. So that's sum over all paths, each weighted by its path probability. Okay? So to do that, we basically replaced the Viterbi variable with the forward variable. And the forward variable, instead of saying, oh, I'm going to recursively compute the maximum by choosing the maximum over all previous maxima, now we're recursively computing the sum by summing over all previous sums. Okay? So basically, again, we're updating the sum using this emission probability times the sum of all previous total probabilities, the sum of all probability mass going through the previous states, each scaled by that transition from the previous state to the current state. Okay, so let's see who's with me on the forward probability score, which is defined exactly the same way as Viterbi, but now we're taking the sum at every position, and then we're summing again through the whole matrix the total probability, basically adding up all of the possible paths systematically. All right, so 14, 9, 2, 1, 0. Okay, so then the forward algorithm could be computed in the same way, column by column, and at the end we just sum up all these numbers and we sort of end up with a sum with a maximum. Of course, doing this in log space is the only option. Otherwise, the numbers become infinitesimally small. So what we're going to be doing is that we're going to be summing log scores, and therefore we're going to be approximating with a particular approximation. Okay. All right. So we saw a lot of algorithms. Now we're going to switch to talk about how do we increase the state space? How do we add additional memory? So we're going to step back and sort of say, great, this very simple HMM was nice and cute, but let's look at the diversity of HMMs. And then we're going to switch to posterior decoding, and then we're going to talk about learning, and then we're going to talk about conditional random fields. Again, this is the optional part of the lecture to basically get you exposed to, a much more powerful probabilistic model. But first of all, let's now expand the state space. So basically, the first example of an HMM was really a toy example. It was only 2 states, a very simple emission vector of only 4 characters, and very simple transitions between them. Okay, that was 2 states capturing the different nucleotide compositions, GC rich versus AT rich, and then it was emitting individual nucleotides. Now, let's talk about how to detect dinucleotides. So right now, we want to actually increase the memory of our system. But I've just told you that hidden Markov models and the Markov chains that they're based on are memoryless. So how can we increase the memory of a memoryless system? Okay, so here's what we're going to do. We're going to remember more. We're going to remember something more than simply, am I in a promoter or in a background state? Okay, so again, Markov models are memoryless. In other words, all memory is encoded in the current state that I'm in. To remember additional information, I need to add more states. So if I want to remember the previous character, I could augment this plus state to be plus and A was the previous character, or plus and C was the previous character, or plus and G was the previous character, or plus and T was the previous character. Okay, so the way that I can create more quote unquote memory in my Markov chain is by increasing the number of states. Okay, and we often talk about the state of a computer system by talking about the memory that it has. So it's very appropriate that we're augmenting the number of states to capture more state. So a 2-state hidden Markov model has minimal memory. The 2 states are GC rich versus equal probability. The state, the emissions, all of them, everything only depends on the current state. And the current state only encodes 1 current nucleotide. So to encode dinucleotide frequencies, or trinucleotide frequencies, or dicodon frequencies, we want to expand the number of states. Okay? So here's the idea. The idea is that instead of having a simple HMM with only 2 states, I'm going to now have an 8-state HMM. There's going to be 4 promoter states and 4 background states. Okay? And then these should be either background or promoter for plus or minus. So the memory of the previous nucleotide will be encoded in the current state. So with GCRH, I'm going to have 4 states, and background, I'm going to have 4 states. And the memory can encode either the previous character or even the current character. So if the memory encodes the current character, that basically means that each of those can emit only that character, but the transition probabilities, in fact, capture the dinucleotide frequencies. So that's one solution. The other solution is that the state encodes the previous character, and therefore each state can emit any of the 4 characters. Okay? So we basically now have a single model that combines 2 Markov chains, each with 4 nucleotides. We're going to have the promoter states, or plus, with ACGT, which emit symbols ACGT in CPG islands, and states ACGT for the minus for outside CPG islands, where the emission probabilities are going to be distinct for the plus and the minus states. And we're going to infer the most likely set of states giving rise to the observed emissions. And therefore, we're going to be painting the sequence with, again, plus and minus states. So the reason why we need so many states is that in our simple GC content example, we only have 2 states, and we need 8 states here because we're encoding the memory of the previous nucleotide. And we're encoding these nucleotide transitions, and therefore the dinucleotide nature of CPG islands. So why do we call them CPG islands? We call them CPG islands because they basically have on the phosphate backbone, they have a C base, and then another phosphate, and then a G base, and another phosphate. So the P denotes the phosphate backbone in order not to be confused with the fact that GC base pairs are connecting on the opposite strand of the double helix. Okay, so we're going to now count dinucleotide frequencies, and we're going to have 16 possible dinucleotides, and therefore 16 transition parameters between these dinucleotide frequencies. And alternatively, as I mentioned, we can have 16 states, each emitting a dinucleotide. So every state could be emitting GA, GC, etc. So what we're going to learn is two different Markov chains. The plus model is going to be the CPG model, where C followed by G is actually very frequent. And the second one is the CPG minus model, where C followed by G is much less frequent. So the transition probabilities for each model are going to be encoding the differences in dinucleotide frequencies. And why are CPG islands important? Because these CPG dinucleotides can be methylated on this side, but also on that side. So the methyl C can be methylated on both sides, and that is a mark of repression. So when CPG is methylated across evolutionary time, it often loses the methionine, and the base, the methyl C base, is actually converted into a T. So you basically have these CPG to either TPG or CPA transition, depending on whether the C on one strand or the C in the other strand was converted into a T. Okay, so CPG on the forward strand is CPG on the reverse strand, but if it deaminates, the forward strand becomes a TPG. If it deaminates the backward strand, it becomes a CPA. Okay. So that's why CPG islands are important, because they are capturing the methylation process of promoters. And methylated DNA tends to lose this CPG and become TPG. And that happens throughout the genome. Most of the human genome is very heavily methylated, except for a few promoter regions that are highly unmethylated, and a few enhancer regions that are dynamically hemimethylated or intermediate methylated. So if you basically look for CPG islands in the human genome, that tells you all of the places where promoter regions are devoid of any kind of methylation historically through evolutionary history of humans, and therefore more likely to be unmethylated regions, unrepressed regions, and therefore more likely to be promoter regions. Okay, so that's what CPG islands are capturing that's different from GC richness. So they're capturing the fact that the C is followed by a G. Okay, so I'm going to do two polls. Number one, who's with me on the biology of why we care about CPG islands and what C followed by G with a phosphate in the middle actually means, and why these correspond to promoter regions, which are generally unmethylated. Okay, so there's a little bit of confusion on the biology. Only 14 people are at full understanding, 7 intermediate, 2 in the middle, and then 6 are below the middle. All right, so that's for the biology. Now on the computer science part, who's with me on augmenting the state of our HMM model, and then including 4 states for each of the plus and the minus to remember the previous nucleotide? So who's with me on the computational part of augmenting the state? Awesome. So 12, 14, 1, 2. Okay. So are there any questions so far on sort of dinucleotide encoding and how we can use these sort of 4 states here? Okay, so this is the sort of simplest possible model. So we basically have GC-rich regions and then CPG-rich regions. So here we have 8 states, 4 states each for plus or minus CPG island with different transition probabilities. And then these capture CPG poor versus CPG rich regions. And then this is emitting dinucleotides. Again, the very first column is detecting conserved regions with 2 states, conserved, not conserved, and the overall level of conservation. Let's now get into protein coding genes. So with protein coding genes, we want to actually have a little bit more information, a little bit more versatility. So let's introduce a little bit the process through which a protein coding gene becomes a protein. So you basically have, for example, for this human gene, for casein kinase 2 subunit beta, you basically have this ATG, that's the start codon. And then you basically code a protein over all the green characters. And then you skip the black part. And then you continue making a protein for the green characters and you skip that black part. And then you continue here and you skip the black part. You only sort of concatenate the green together. And in the end, you basically have a stop codon, TGA or TAG or TAA, which basically tells the ribosome to stop translating. So when the genome is transcribed into RNA, before it gets translated, that's when splicing happens, where you can see the splice sites are starting with GT, a long sequence, AG. 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 Okay. So, and here those two shouldn't have been highlighted. So there's basically these very simple rules that allow you to sort of go from the DNA transcription into the pre-mRNA to become the mature mRNA after splicing. So basically splicing relies on these sequences. And as we write computer programs to go and find where are the splice sites, what we need to do is remember the codon composition properties of the every exon. So in green are the exons, and in black, in between the exons, you have the introns, and then here you have the untranslated region, or the UTR, the 5' UTR and the 3' UTR. Okay, so now let's see how many states do I need in my hidden Markov model to be able to capture the transition into a gene. Maybe I want a state here that captures the mRNA 3' UTR. Then I want to transition through an ATG. So I need a state encoding the ATG. Then I need to remember that I'm in an encoding region and therefore emit trinucleotides rather than mononucleotides. Then I need to sort of recognize this splice site, the acceptor and the donor. So basically the donor is GT, the acceptor is AG, or CAG, which is the most common consensus. So you basically have sort of donor acceptor, donor acceptor, donor acceptor. And then that basically means that I need to sort of have a start and first exon, and then intron, exon, intron, exon, intron, exon, intron, exon, intron, exon. How many exons can I have? Any number. So that basically means that I can encode this in 2 different states, one for intron, one for exon. And every time I transition from intron to exon, I go through a splice site. Okay? Who's with me so far on sort of this more complex hidden Markov model for capturing the structure of protein coding genes with start codons, stop codons, introns, exons, splice sites to transition between them, and so on and so forth. Awesome. So if you're with me so far, this is nice and awesome. Now there's going to be a little bit more complication, okay? So notice that the reading frame matters. If I find a stop codon in the middle of this region, it better be a stop codon in the alternate reading frame. So for example, here I see a TGA. If that TGA is in frame, the ribosome is just going to stop there. So what we need to do is remember what reading frame we're in when transitioning from one exon to the next exon. And from the end of this exon to the beginning of the next exon, I have to actually remember the reading frame of translation, okay? Because every triplet is one codon and becomes one amino acid. As I go through, I need to actually remember that. Who's with me on this subtlety here, that I actually have to somehow encode the memory of, am I on the first codon position, the second codon position, or the third codon position? Okay, so there's some folks like who are really, really with me. So basically 85% of you are very well with me. And then there's 4 people who are not 100% with me yet. So let's talk a little bit more about this. So there's a big difference if I basically start translating this every 3 nucleotides. And if I now have 2 nucleotides here overhanging, I better have 1 more nucleotide here as part of the previous codon before I sort of continue into this, okay? So basically, if I have 2 green characters here, I better have 1 more green character there to effectively complete that codon. And here, if I have 1 green character, I better have 2 more green characters to complete that codon. And here, if I have fully completed a codon, I better start a full codon here. And here, if I have, 1 character, I better take 2 more green characters from the next one. So that basically means that while I'm here, I have to remember that I still have a 1, 1, 1, 1, 1 overhang. Here, I have a 2 overhang. Here I have 1 to go. Here I have 2 to go. Here I have 0 to go. Here I have 2 to go, and so on and so forth, okay? And then, for example, here, I might have 2 more to go. The splicing machinery couldn't care less whether you stop in the middle of the splice machinery couldn't care less if you're, in the middle of a codon when you have your splice site, because after splicing, the translation will, be oblivious to how you had stopped. But when we write, as computer scientists, when we write a parser for the mRNA recognition of a protein coding gene, the interpretation of that sequence in this reading frame is dramatically different from the interpretation of the same sequence in a different reading frame. So basically, if I parse this exon in the wrong reading frame, I'm going to make all the wrong inferences about it. All right, so let's see now who's with me so far and sort of how I need to encode this memory from one codon to the next codon, from one exon to the next exon. Okay, so I have nearly everyone with me here. That's awesome. Okay, so now let's see how we can actually do all that in in our hidden Markov model framework. So basically, what we're going to have is first some exons in the 5' UTR, then a start codon. Then we're going to be going through donor, acceptor, GTAG, exon, intron, exon, intron, exon. And eventually, we're going to be at a terminal exon. And then we're going to go back to this untranslated region, now on the 3' end. Okay? So this is going to be our state transitions in a hidden Markov model. Okay? So in practice, this is what these actually look like. So this is basically the actual architecture of a protein coding gene detector that basically says, I'm going to look for a Tata box, then a 5' UTR, then a start codon, then I'm going to enter the initial exon, then I'm going to give a donor, and then I remember that I'm in a offset 0, offset 1, offset 2. I need to have 3 intron states, and then depending on how I enter my exon, I will then exit into, again, an overhang of 1, 2, or 0, depending on how I exit. So I can go through a bunch of initial exon, a single initial exon, and then a bunch of internal intron, exon, intron, exon, intron, exon, until the final exon, and then the stop codon, and then the 3' UTR, and then this is the transcription termination signal, and then back to any character, which we denote as N in genomics in general. Or I might just start right away with an ATG without detecting the Tata. And instead of going through exon intron, I might also just have a single exon and then the stop, and then either the 3' UTR or finish right away. Okay? Who's 100% with me on this one? Let's see. Pretty awesome. So we have 11, 13, 2, 3. And now, who can tell me what's going on here? Here it says plus strand. What should I put in the minus strand? So use your chat function if you can think of an answer, or just raise your hand, or just simply unmute yourself and speak up. So what's going on here? So all of this is basically detecting, you know, start, exon, intron, exon, intron, exon, intron, exon, intron, stop. What do I need on the other side? So what I need on the other side is the ability to detect genes that are encoded on the negative strand. Because in DNA, you basically have the ability to encode the genes both in the forward strand and the reverse strand. And if you only have a single-stranded predictor, that basically means that you might misinterpret a gene that's actually truly on the opposite strand as being on the forward strand. So that's why instead of just building a plus strand predictor only and then running it twice, what you're going to be doing is actually building a joint model that has, you know, both forward and reverse strand predictions simultaneously in order to be able to, you know, detect negative strand genes without sort of being confused by their shadow on the positive strand. Because the trinucleotide composition pattern of a gene actually leaves an imprint on the opposite strand from where it sits. Okay? So we have gene versus intergenic, we have start codons, stop codons, we have 3' and 5' UTRs, untranslated regions, exons and introns, and then we're remembering the reading frame with E0, E1, E2, and then the sequence patterns to the transitions between them, their ATG and then TAG. You can see here we have to go through ATG, we have to go through TAG. Okay? So basically the reason why I'm showing you this is for you to realize that hidden Markov models are not just cool algorithmically, but they're extremely versatile in the architectures that you can sort of encode for them. All right, I see a message in the chat window. What's the issue with just running a single-strand HMM on both strands? So also, what's the issue with just looking for ATAT in addition to TATA and TTATT in addition to AATAA to deal with this? So yeah, basically what you need to do is having exactly that on the opposite strand to be able to, so the reason for that is that the trinucleotide composition on the forward strand is, as I was saying earlier, leaving a shadow. Let me see, you know, if I can show you easily what that means. So that basically means that if I am, so let's see, chat, you might have one state sequence that has genes on both plus and minus strands. Yes, absolutely. You can have genes within genes, and so basically, you know, it's anything really goes. If you can imagine it, nature has probably done it like a dozen times. So if I'm interpreting a sequence on the forward strand every 3 nucleotides, its shadow on the reverse strand is actually looking very similar. So the third codon positions align, and then the codon frequencies are non-random in both strands. So basically, you know, the third codon position here is actually showing very similar signatures to the third codon position here. So as you start sort of building these models, you will realize that there's actually false positives, and some of those false positives come from these shadows on the opposite strand. Let's see, that's my reason for not wanting to run a single strand HMM on both strands. You might have one state sequence that has genes on both plus and minus strands. Yeah, that's exactly right. So basically, you could basically first run it once and then say, okay, there's a gene here, there's another gene here, and there's another gene here, and there's another gene here, and then run it again and say, oh, cool, there's a gene here, there's a gene here, there's a gene here, and there's a gene here. That could be one way to go. But chances are, when you run it forward, it'll basically say there's a gene here, there's a gene here, and there's some kind of weird gene here, and there's like another weird gene here, and oh, and there seems to be another weird gene. Like basically, for each of the reverse strand genes, you're going to find some weird signatures on the forward strand, and the reason for that is, you know, that it looks non-random, so it doesn't get captured very well with your HMM. All right, so that's sort of, you know, maybe one of the most complex HMMs in genomics, so basically looking for protein coding genes. But we can also use HMMs with less complex transitions, but more complex states. So for example, you know, my own postdoc, Jason Ernst, developed this chromatin state hidden Markov model for detecting different epigenomic signatures using their chromatin mark combinations, and therefore infer in the hidden states enhancers, promoters, transcribe regions, repress regions, repetitive regions, based on the vector of chromatin mark frequencies in each of those in the genome. So this is what this Chrom HMM model looks like, that basically learns these hidden Markov models. So this was initially published in Nature Biotech as a concept, then in Nature as the first application, and then in Nature Methods as the software package. And it's now being used by, you know, hundreds of labs across the world. So what Jason basically did is develop a hidden Markov model with 50 different chromatin states, each of which emits a different vector of histomodification marks. And you can see that the model was able to learn a set of states that have a lot of these you know, marks here, and then another set of states that have different sets of marks, different sets of marks, different set of marks, and so on and so forth. And then this is the transition matrix between them that basically captures the spatial relationships of these states. So this has now been used in countless applications to basically understand the chromatin landscape in many different species. And we're going to talk about this model in the corresponding epigenomics lecture. Okay? But I wanted to sort of lead you with this, and also to detect protein coding conservation. We've basically also built a two-state hidden Markov model that basically captures different evolutionary signatures of those states to distinguish protein coding exons from non-coding exons based on a 64 by 64 matrix of codon substitution frequencies. And we're going to talk about this in the comparative genomics lecture, and we're going to talk about this in the epigenomics lecture. Okay? So this was all about increasing the state space, basically finding GC-rich regions versus finding CPG islands, and then gene structures with Genscan, and then chromatin with ChromHMM, but also conservation with Congo, and so on and so forth. Okay? So now let's talk about posterior decoding, and then we're going to talk about learning. Okay? So, but first of all, who's with me so far on this whole matrix here? Do you guys see sort of how we can augment these HMMs with a lot more complexity? Okay, so 9, 15, 0, 1, 0. So basically nearly everyone is well above average. All right, so let's now switch to posterior decoding. So remember our matrix here. We talked about scoring a particular sequence given a single path and scoring a particular sequence given all paths. We also talked about decoding a particular sequence, X, taking the maximum over all possible paths, and then returning the most likely path of our sequence. But there's another way to decode, which is posterior decoding, and that basically gives you the path which contains the most likely state at any time point. Let me try to explain. So if you're sort of parsing through your HMM and you basically say, okay, I'm an exon here, I'm an intron, I'm a transition, you know, now I'm really an exon, then now I'm in a transition, and now I'm in a, you know, so basically these are the states, these are the K states over time, okay? Is everybody with me here? So can everybody sort of understand what I'm drawing? So basically what I'm drawing now is the probabilistic parse that basically tells me what state K am I at each time point, okay? Beautiful. Great. So what this allows me to do is now start parsing, you know, not just one path, but multiple paths. So 14, 6, 3, 0, 0. So this is one path. Here's another path. So I'm going to draw the next one in blue. So this path basically chooses this other state here, but then goes back to the same state and maybe transitions a little bit further. And then maybe it goes through the exact same transition here. And maybe here it'll just choose another state for a little bit and then make the same transition, choose another transition here, and then finish the same way, okay? That's one parse in red. That's another parse in green. I can do a third parse of the same sequence. Maybe the third parse will sort of agree with the red one for, you know, parts of the sequence, and then maybe agree with the blue one for other parts of the sequence, okay? So I want you to sort of conceptualize the multiple ways that I can interpret my sequence, and then start asking what is the most likely state at a particular location. So now I have a third path, a third parse through my sequence. And now I can basically say, if I take this position here, what state am I in? And if I take this position here, what state am I in? And all of these black locations are pretty confident. They all agree that, I mean, you know, this state here, for example, I'm an exon in this particular case, okay? So basically, this is the exon state, and then that's also the exon state, and that's clearly an intron state, and that's clearly a 3' UTR state, okay? However, there are other places where there's more uncertainty. So for example, if I ask you, what state am I in here? It's possible that the most likely path, let's say, you know, this red path here, is actually the Viterbi decoding max likelihood path. Okay. So what the Viterbi path will basically tell me is that, oh, at position, you know, I don't know, 37 of my sequence, I am in a exonic state. That's what the Viterbi path will tell me. But now, if I look at all other paths seem to disagree. They're all saying, no, 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 you're in an intron state here. You're clearly not an exon. It's only the maximum likelihood path that basically told me that I'm in an intron here, or sorry, that I'm in an exon here versus an intron. And what posterior decoding tries to do, it basically says, can I find not just the maximum likelihood path, but the most likely state at every position, taking the sum of all of the probabilities summed over all paths, okay? So let's do a quick poll here and see who's with me. So basically, what Viterbi decoding does is that it tells me what is the state at every position that the best path, that the maximum likelihood path went through at that position. That's Viterbi decoding. What posterior decoding tells me is what is the state with maximum total probability summed over all paths at every location. Okay? So you guys are awesome. This is great. So 11, 12, 1, 1, 0. Actually, I'm going to do one more poll, which is who feels that there's a cool new concept here that this posterior decoding is giving you. So basically, posterior decoding is a very different type of interpretation. It sort of helps you think about all of the paths through a sequence rather than just the maximum likelihood path. And the reason for that is that the maximum likelihood path might contain only a small fraction of the total probability through all of the paths. And that maximum likelihood path might not be the best interpretation at any one location. And that's what posterior decoding tries to do. It tries to sort of, you know, overcome that. So 16, 7, 1, 0, 1. Okay. So now let me go back to the lecture and now enter this second, you know, matrix, the second column of the matrix. So before we had the one path column, and now we have the all paths column. We already saw how we can sum over all paths. So now here's a very weird notation, but I hope it'll make sense. So pi hat is the path consisting of the pi I at every position, such that this pi I maximizes summing over this pi I chooses the state k, which maximizes the total sum probability of all of the paths that parse x and go through the state k. Okay, so it basically says of all of the possible paths that are traversing my sequence, how many are going through state k at that position, at that particular position. Okay. So this is the path that contains the most likely state at every time point. Okay. So in other words, what we want to do is sum over all paths that go through the sequence and go through this state at that position, and then all of the paths going out of that sequence. So what is the total sum probability of all of the paths ending here? There's a question. Just in general, I'm sorry, wrong person. So what is, you know, I've already showed you how to calculate the total sum probability ending at that location here. What do we call this? We had a name for this. This is not Viterbi decoding. This wasn't the Viterbi variable. It was the forward algorithm that was giving us the forward variable. So basically, I already know how to compute all of this summed ending up here. Okay, so so basically, in order to calculate the posterior probability of being in state B at that location, I can sum over all of the paths and end in B. But then I also want to do the sum of all of the outgoing paths. So what we're going to do here is the same trick that we used for the recursive algorithm that allowed us to use dynamic programming with linear space by sort of computing the same path, but now backwards. So for the forward algorithm, I can compute the total forward probability trivially from the left to the right, but I can also compute P of X from the right to the left using the creatively named backward algorithm. So I can calculate basically the forward algorithm all the way to here and the backward algorithm all the way to here. And if I sum them up, that basically gives me the total probability of going through this particular state. So who's with me so far on understanding that to get that total probability, I already have the forward algorithm. All I need to do is compute the backward algorithm and have them meet in the middle. And that basically gives me the sum of all of the paths going into one state and the sum of all of the paths exiting that state that I calculate going backwards from the right to the left. Okay, so yeah, 90% of the folks are 60% and above, which is awesome. And then 2 folks are below. Okay, so that's the most, so basically there's many ways to sort of say what is the most probable label at a single position. What I could do is simply calculate, again, the most probable label at each position, doing this for all of the positions, and then there's multiple settings in which I can do this. One setting is I haven't observed anything. And there, I just simply ask, well, which of the 2 states is most likely a priori? Is it promoter or background? And that's just come from the stationary distribution of the HMM, of the Markov chain. Not very interesting. So if I've observed nothing, I just use the prior information. If I've observed only the character at that position, then it's basically my prior and the emission probability. So remember this whole sort of reversing the directionality. I have the probability of the data given the hypothesis. I can use that to infer the probability of the hypothesis given the data divided by the probability, the prior probability of the hypothesis, okay? And that gives me the posterior. And if I've observed the entire sequence, that's when posterior decoding comes in, okay? And I want you to build a little bit of intuition here. The intuition is the following. If I've observed the beginning of the sequence and I end up with B being, I don't know, very high probability, how can anything else after this change my inference for how likely is it that B is actually the most likely state? It's a little counterintuitive, right? You can just like, okay, great, I'm parsing, I end up in B, and then it sort of, you know, continues out. Why would the rest have any impact on B, on this particular state? The reason is that if I don't know, have GC, AAA, and then T, AA, etc., then this actually increases the probability that this is a background region. If instead I have GC, AA, and then GC, CG, etc., then that basically dramatically increases the probability that those 2 characters, G and C, are going to be parsed as a promoter region along with the other chunk of really high GC rich characters. Okay, so who's with me on the intuition for why observing additional sequence afterwards can actually have an impact and change the current interpretation? The reason is that it changes the overall probability of all of the paths, and therefore it can sort of directly influence this. Okay, so 12, 11, 0, 0, 0, that's great. Okay, so with no knowledge, no characters, it's simply the time spent in the Markov chain states, and then the most likely state, the prior. With very little knowledge, just that character, it's the time spent, adjusted for the different emission probabilities, and we can use Bayes' rule to change the inference directionality and basically infer the probability of the path being in state K given a character, which is basically the probability of being in seeing that character from that state, you know, times the prior of that state divided by the total probability of observing that character. And if I have knowledge of the entire sequence, all of the characters, then this is summing over all paths that emit G at position 7, and that's posterior decoding. Okay? For those of you who prefer to derive it algebraically, so basically, in order to compute the probability of that sequence, given that we're in this state, given the entire sequence, we basically want to compute it all the way down to this path here, being in state K. And then from here, I want to compute that backwards using the backward algorithm. Okay? And then when they meet in the middle, that's when I can sort of calculate that. So the backward algorithm basically can be expressed as a function of itself, and then sort of we can infer the backward algorithm at position I as a function of all of the backward algorithms at position I plus 1. And notice that we're moving backwards now. Basically, we're calculating the lower index as a function of the higher index. And notice that also the emission is now factored into the equation rather than before where we were taking it out. And the reason for that is that we want things to add up with everything taken into account. And that's why the emission probability corresponds to the next state rather than the current state. Okay? So we can define again the backward algorithm recursively based on it being computed for all of the higher indices. I can compute the lower indices. And then notice that the next emission is now inside the sum rather than outside the sum, because it actually depends on the next state rather than the current state K. So then we can run the backward algorithm from right to left, and then we can put it all together by computing one matrix with a forward algorithm, one matrix with a backward algorithm from right to left, one for the forward algorithm from left to right. And then what posterior decoding is simply the sum of the forward variable and the backward variable at that location. So the probability of being in state K is the forward vector plus the backward vector divided by the total probability. So it's the probability of being in the ith state in position K given all the emissions. So it's the total product, it's the sum of the logs or the product of the probabilities. So with posterior decoding, we want to find the most likely state, and then the posterior decoding quote-unquote path is very useful for classification, and it's more informative than the Viterbi path by star. And it's a more refined measure of which hidden states generated X at every location. However, it may give an invalid sequence of states. Not every J to K transition may be possible, okay? And now, let me go back to my diagram from earlier. Perhaps the reason why the maximum likelihood Viterbi path gave us this as an exon rather than as an intron, perhaps is because, you know, the transition from the previous time point to the next time point would simply not be possible in the, you know, maximum likelihood path, because it requires that these probabilities are non-random, are non-zero. But, you know, for example, posterior decoding might tell me that here I have an intron, and then I have a stop codon immediately thereafter, right? And that's nonsensical, because in my HMM for Genscan, I saw that, well, the intron has to first go to terminal exon and then go to a stop codon, okay? So, you know, the fact that there's no intermediate state E is problematic. And that's sort of, you know, one reason why the Viterbi path might actually be better in some cases. And the reason for that is that it always gives you a sensible path. But the posterior decoding gives you, you know, higher per position probability. All right, so we talked about posterior decoding and finding the most likely state over all paths. So basically, we've now covered the first two rows of our matrix. We've talked about scoring over one path, about decoding over one path, about scoring over all paths, and then posterior decoding over all paths. Okay, so Who's with me so far for the first 4 quadrants of this matrix? Okay, so 11, 12, 1, 0, 0. And now, how's the pace so far? Do you guys feel like we're going too fast, too slow, or just right? Okay. Wow, so 16 just right, 6 above, 3 below. So maybe just a tidbit too fast. Okay, so now let's talk about learning. So remember when we talked very, very early on about these 3 components. So basically, we now have the ability to emit DNA sequences of a certain type. We have the ability to recognize DNA sequences of a certain type. How do we now learn the distinguishing characteristics of each state? How do we train our generative models on large data sets? And how do we learn to classify both labeled and unlabeled data? Okay, so the way we're going to do that is build it up. So first, we're going to basically look at supervised learning. Okay, so first we're going to look at supervised learning. If somebody comes and basically says, here's an annotated large sequence, how do you find the emission probability vectors and the transition probability matrix given a labeled sequence? And then how do we do this unsupervised? Given only a large sequence, how do we figure out both the annotation and the parameters? That's sort of the magic part, okay? But the supervised part is kind of boring. It's super, super easy. So let's look at supervised first, the super easy way, namely Viterbi decoding. And then, supervised learning if everything is known, and then an easy unsupervised learning, and then the much more powerful Baum Welch expectation maximization, unsupervised learning over all possible paths. Okay, so now let's focus on the left side and look at supervised first. Okay, so how do you train your HMM? What you would like to have is estimations for what is the probability of observing each character given that I'm background. What is the probability of estimating each character given, of emitting each character given that I'm promoter? And what is the transition probabilities between B to B, B to P, P to B, and P to P? Okay. So this is actually quite simple. If I'm given an annotation of my sequence, if I'm basically told that this is my sequence and this is the annotation, then I can infer the most likely parameters for emission probabilities just by counting. How many times was I in the background state? 5. How many times did I emit a G from the background state? 2. What is the maximum likelihood estimate of the emission probability of the character G from B? I expect all of you guys to type frantically in the chat window. So I observed G 2 times out of 5. That basically means that the probability of emitting a G from the background state is basically 2 out of 5. And then the probability of emitting a C is also 2 out of 5. Probability of emitting an A is 1 out of 5. Okay? Who's 100% with me on the emission probabilities here? So, oops, sorry, wrong poll. So basically, if I have an annotation, my maximum likelihood estimates are just simply the fraction of time that I was in the background state where I emitted each of the 4 characters. And then the exact same thing for my probability of emissions from the promoter state. Okay? So 19, 4, 1, 0, 0. This is awesome. So now let's look at the transition probabilities. We can now ask how often, what is my transition probability between background and promoter. So I mean background 1, 2, 3, 4, 5 times, and a transition promoter 1 out of the 5 times. So that's 1 out of 5. What is my transition probability between promoter and promoter? I mean promoter 3 times and a transition promoter 2 of the 3 times. So that's 2/3. What is my B to B probability? 1, 2, 3, 4, 5 times. I self-transition 3 of the 5 times. Okay? So this is the most trivial thing. So if I have an annotation, I can infer both the emission parameters and the transition parameters super easy. Okay? So there's one caveat, of course, that, well, we don't want zeros, because a 0 probability is an infinite penalty in log space. So what we're going to be doing is simply avoiding overfitting by eliminating the 0 probabilities by adding some pseudo-counts. Okay? So basically, pseudo-counts is, you know, just making sure that the probabilities are non-zero. And if you include just an epsilon pseudo-count, you're just avoiding the 0. If you include larger pseudo-counts, it's basically just like a strong prior belief. So you could basically say, well, I'm going to start with 2/5 because that's sort of what I'm expecting. That's sort of my prior belief. And with, again, Bayesian statistics, your prior belief could start here, and after a lot of observation, you might actually move here. When with the frequentist approach, you might say, oh, this is exactly my estimate. This is exactly 2/5. Whereas the Bayesian approach might say, well, my prior is a Gaussian centered around 1/5, but I'm observing something higher. So, you know, I'm going to move my Gaussian estimate a little higher to maybe 2/5, as opposed to sort of, that's exactly what the frequentist approach would give. Okay. All right, so that's for pseudo-counts, and basically the way that we obtained these numbers when I showed you earlier for CPG islands was simply counting how many times did I have the letter T followed, you know, by the letter S inside CPG islands versus outside CPG islands, and then that's sort of what gave us those numbers. Okay, so that's supervised learning, and that's super true. Unsupervised learning is a little harder, so let's dive into it. So how do we infer, given only the sequence, both the annotation and the parameters? Well, we don't really know what to count. We don't really know where to start. If I had an annotation, then I could infer the parameters. That would be supervised learning. If I had the parameters, I could infer an annotation. That's just Viterbi decoding. You know, that's the middle left of my matrix. But if I have neither, how do we solve it? Well, one way to do it is to imagine that we start with some parameters. And then, if I start with some random parameters, I can go ahead and calculate the most likely path given those parameters. And then, once I've calculated the path, I could re-estimate my parameters from the path. And if I have an estimation of the path again, I can basically, if I have an estimate of my path, I can basically infer the maximum likelihood parameters that are best for that path. And then once I have the new maximum likelihood parameters, I can recalculate an estimate of my parse of my path and provide a slightly different annotation, and then re-estimate the parameters, and then continue until convergence. Okay, so why is this even working? The reason why it's working is because if you look at the your sequence itself, it might actually have some regions that are very high GC, and then long space in between, and other regions that are very high GC and long space in between. Or it might tell you that, oh, well, this region is, I don't know, has very weird trinucleotide frequencies, and this other region has very weird trinucleotide frequencies. Even if you start with a random initialization, you might end up with, some parse here that partly captures it, and another parse here that partly captures it. But the beauty of it is that you're driven still by the genomic sequence. So at the next iteration, when you sort of go with your blue parsing, you might sort of capture more of that. And then here you might capture more of that. And at the next iteration, you might again capture even more of that and even more of that. So even though you're starting from a random set of parameters, at every iteration, you're using the sequence and the unique, intricate properties of the sequence when you're applying those parameters to parse your sequence. And even if you dreamt up some really crappy parameters that just don't match the sequence at all, as long as they initially, just like the green guy here, capture some aspect of the particularity of your sequence, they will move towards that local maximum, and they will capture more and more and more of it to the point that they will ultimately learn the particularities of your underlying sequence, and they will converge to sort of capturing the fact that there's two distinct nucleotide composition properties of the genome overall. All right, so let's see who's with me so far on both the logistics of unsupervised learning and the intuition behind unsupervised learning, and how, in fact, the sequence itself is guiding, it's constraining the set of all possible parameters that you could go through. It's constraining them to the set of parameters that are truly capturing the properties of your sequence. Awesome. So 12, 10, 3, 0, 0. This is really great. All right. So that's with the simple, quote unquote, unsupervised learning. So basically, the idea of this simple answer is you estimate the best guess of the transition and the emission parameters, and then you update the probabilistic parse of your sequence based on these parameters, and you keep repeating. And we're going to look at two different settings. The simple setting is going to be Viterbi training, where the best guess here is going to be the best path, okay? And then we're going to look at the fancier version, which is expectation maximization, where the best guess is actually all paths, each weighted by its probability, okay? So Viterbi training is basically what we talked about already. So you just run the Viterbi algorithm and you get your parse, and that's it. At every time point, you just have one emission and one transition. So then it's super trivial to count. It's as if somebody had given you the right answer. But with the all paths view, which is the bottom right of this matrix, the all paths view is a little more complex. We know how to do posterior decoding. That's easy. But what we want is not just the posterior decoding path, because even that posterior decoding path gives us only one interpretation of the sequence at every position. Instead, what we want is all interpretations and all paths, not just one path, the posterior decoding path. So what we're going to be doing is summing over all paths this total probability, and then maximizing the set of parameters, summing over all those paths. So the basic idea is expectation maximization. We're going to use the model to estimate the missing data in the E step. That's sort of estimating an annotation of our sequence. And then we're using that estimate to update the maximum likelihood parameters of the model. And that sort of expectation of the parse, maximization of the parameters, estimation of the parse, maximization of the parameters, okay? So what EM does is it estimates the expected probability of the hidden labels given the current, the latest parameters, and the observed unchanging sequence. The unchanging is extremely important because it keeps guiding our parse. And then the M step is choosing new maximum likelihood parameters over the probability distribution given the current probabilistic label assignments, not deterministic, not the maximum likelihood path, but the probabilistic estimate of all of the paths. And that is guaranteed to increase at every iteration. So let's get down to it. So how do we estimate the total probability of emitting A from character B, from state B? It's super trivial. We basically calculate the forward algorithm and the reverse algorithm, the forward and the backward algorithm at every position. And then my estimate, my maximum a posteriori estimate of my parameters of emitting character B from state K is going to be, of course, normalizing by the total forward sum at the end or backward sum at the beginning, dividing by 1 over px, and that's given by the forward order backward algorithm. And then we're going to be summing for all i's where the observation at position I is B. Super trivial. And what are we going to be summing? The forward and the backward algorithm at that location. Okay, let's see. This is super crazy complicated, but if you guys are following, I will be very, very happy. So this is basically calculating the total emission of character A from state background, which is at every position where I had an A, basically go through and calculate just add up the two vectors, you know, in log space or multiply them in regular space of the forward and the backward algorithm at each of those places where there was an A. And then that gives me the sum over all paths of how much probability did I have going to that state. Awesome. So 38710, that's great. Then the transitions are just a tidbit more complicated. The transitions is basically the total probability of ending at state B and transitioning to state P at every offset, every time point. So for every I, so summing over all i's, I'm going to be summing the total probability of ending at that state and the total probability of leaving from that state. What is that? This is simply the forward probability vector here, the backward probability vector here. So it's forward all the way to state K at position I, and backward all the way to state L backwards at position I plus 1. And then in between them, it's just the transition probability and then the emission probability. So transition from K to L, and that's simply the same exact AKL everywhere. And then the emission of whatever character is at position I plus 1 from state L. Okay, let's see who's with me on this one. So this is basically the sum of all emissions and then the sum over all transitions, all of them probabilistic, using the forward and the backward vectors. The reason why this is so powerful is that I only need to compute the forward and the backward vector once. And once I've computed them, I can just reuse them for all these crazy, super complicated algorithms, each time summing over an exponential number of paths, but reading them out through having already solved all of the subparts for each of those. Okay, we have 20%, 30%, 37%, 11%, and then 0%. So most people are sort of, you know, somewhere in the middle. All right, and then this is the derivation of how we can calculate these transition probabilities as a function of themselves. And then that's how you end up with this forward, the transition, the emission, and then the backward. Okay? So we've basically covered all of the algorithms of hidden Markov models right now, of HMMs. We've looked at scoring from both sides now, from one path and from all paths. We've looked at decoding from one path and from all paths. And we've looked at learning using one path, and that path could simply be supervised, where we're given the path, or unsupervised, where we're inferring the Viterbi path each time, or unsupervised over all paths, where we're using Baum Welch training, the full expectation maximization framework, over all paths. Okay? So we basically reviewed the basics and the 3 algorithms. We increased the state space. We looked at learning, and then I guess we're not going to cover conditional random fields. We need to know the number of states, though, for BW. I'm not sure I understand your question, Guillaume. Know the number of states? Yes, of course. So, of course. So the number of states is a user-specified parameter. When we talk about the epigenomics lecture, we're going to look at how we can use Bayesian information criterion, or how we can use 2 steps, one to learn a super complex model and then prune it down, and all kinds of other techniques for inferring what is the model complexity. But given a particular model complexity, then these algorithms allow you to infer the parameters. To decide on what we call hyperparameters, namely the higher level parameters of the model, that's when you can either have an outer loop that basically says, what is the total probability of my model, penalized for complexity, summing over all paths, given my parameters, or other ways of sort of selecting the complexity, the number of states in your model. That's a great question, Guillaume. Any other questions? All right, if not, you're just in time for your next class. Oh, let's see, chat. Oh, thank you. Perfect. And then see you guys on Thursday. We're going to be switching to the next module and talking about expression analysis. All right. Thank you, guys. Bye-bye.
Real quick.